topic, that is regulation of nitric oxide, that is role in agriculture. So, how many of you know about the nitric oxide? Nitric oxide is a fascinating molecule that has gained significant attention in recent years due to its diverse role in both animals and plant physiology. It is not only important for the plant, but it is also equally important for the animal physiology also. And in this presentation, we will explore the biological functions of nitric oxide, its synthesis, regulation in plants and its potential applications in agriculture. So, first of all, moving forward to the discussing the all details, we have to know what is the nitric oxide. So, nitric oxide is a small, highly reactive and diffusible molecule that acts as a signaling agent in biological system. That is why it is very important for the both animals and plants. It was initially considered toxic due to its presence in industrial waste and exhaust fume and associated with the acid rain and air pollution. But however, in the earlier 19th century, researchers discovered its critical role in regulating the human cardiovascular system and leading to the paradigm shift in our understanding of nitric oxide. And for this work in 1992, NO was named as the molecule of the year, and in 1998, 3 American scientists, F. Murad, Fugard and Ignaro, were awarded with the Nobel Prize in the work of NO. So, if we see what are the properties of nitric oxide that making it the molecule of the year. So, the unique properties of NO that make it versatile molecule in biological system, that is its lipophilic, meaning it can easily cross the cell membrane, and it has an unpaired electron that make it highly reactive. As you can see in the structure that NO has a one unpaired electron that making it highly reactive in nature, and this reactivity allows NO to interact with various molecular targets including metals and thiol containing proteins. So, additionally NO react with reactive oxygen species and forming the reactive nitrogen species. So, all of you know the ROS and RNS both are very important and used as signaling molecules at very low level. But when their level is exceed, they are working as a, they create the aging and many processes that called as stress responses. So, at the cellular level, when their condition is at the optimum level, it is very crucial and it work as a signaling molecules. So, NO have emerged as a signaling molecule due to their prominent existence and diverse role in cellular metabolism, signaling pathways and induction of oxidative stress, regulation of stomatal guard cell opening and modulation of plasma membrane fluidity, and contribution in programmed cell death at low concentration. So, it is very important to know at all these processes involved in low concentration. However, if the amount of this NO surpasses a certain threshold level, they have the possibility to trigger the damage to the cell and that causes the cytotoxicity. So, that is why the topic of this my talk is the regulation of nitric oxide, because if it exceed is called reduce the plant cytotoxicity. At lower level it enhances the many biological functions. So, our focus in this talk is on the regulation and beneficial aspect of nitric oxide, not the harmful aspects. That is why I am not going to discuss about the RNS, that is the reactive nitrogen species. I will discuss only the beneficial aspect of nitric oxide. So, if we see the regulation of nitric oxide, there are 3 main pathways that impart the regulation. The first one is the cellular homeostasis, that is achieved by the synthesis and scavenging. And there are some key regulatory mechanisms include the interaction with reactive species and processes like S-nitrosylation and D-nitrosylation, and further NO is degraded by the some enzymes like NO deoxygenase that binds with the, and it also binds with the antioxidant like glutathione. So, that is why antioxidant is also very important for the plants as well as for the human system as well as for the animals, because it binds with the excess oxidants जो भी generate होंगे हमारे body में, that binds with these oxidants. So, if we see the NO homeostasis, it is essential for signaling functions in plants, and the equilibrium between this NO synthesis and scavenging determines the endogenous levels of nitric oxide. which must be carefully balanced to respond to various physiological and environmental stress conditions. And this balance ensures that NO can effectively regulate plant growth, development and stress responses. And for this, uh, first of all, we have to discuss what are the sources and synthesis of nitric oxide in plants, because NO homeostasis depend upon the two things, its synthesis and its scavenging. So, what are the sources of NO in plants? So, in plants, the primary source of nitric oxide is through the reduction of nitrite, that primarily occurring via the enzyme nitrate reductase that located in the cytosol and plasma membrane. With other potential sources include mitochondrial electron transport, xanthine dehydrogenase and the NOS pathway, which was first discovered in animals, but now it is also reported in plant system. And the other pathways of nitrosynthesis, NO synthesis is the non-enzymatic pathway that under the acidic conditions, however the exact mechanism of NO production in plants are still being researched, it says that the under acidic condition NO is simultaneously produced from the nitrate. So, there are the multiple pathways of NO which we are going to discuss one by one. So, this biosynthesis include mainly two pathways. First one, that is the enzymatic, and the second one is the non-enzymatic. There are several enzymes which involved in the synthesis of nitric oxides. So, first of all, what are the biosynthetic pathway that due to the enzymatic activities? So, the key enzyme pathways include the NR pathway, NOS pathway, xanthine oxidase pathway and mitochondrial electron transport chain, and the first one in this the nitrate reductase pathway. This NR reduces the nitrate to nitrite, which can further convert it to NO, and this pathway is crucial at under low oxygen condition. And the second one is the nitric oxide synthase like activity. This enzyme, although a homolog to animal NOS and has not been identified in plants, but its like activity has been reported and utilizes the arginine to produce the nitric oxide in plant system. In similar way, the next enzyme, that is the xanthine oxidase, that generate the nitric oxide through the reduced nitrite to NO under the hypoxic conditions. And the last one in the enzyme activity is the polyamine oxidase pathway. In this, oxidation of polyamines such as spermine and spermidine can lead to the NO production through the reduction pathway. So, if we see, these are the enzymatic pathway, and the second one is the synthesized by the non-enzymatic sources, and these are the basically reduction of nitrite to NO can occur in the presence of reducing agents like ascorbates and phenolics. So, these are the sources of non-enzymatic, and it is also generated at low pH. At acidic pH, nitrite can simultaneously convert it to NO. So, enzymatic pathways involve the enzymes and NO simultaneously generated at acidic condition, where it can also be reduced through the ascorbate or phenolic through the in the non-enzymatic sources. So, if you see the overview of the nitric oxide biosynthesis, this is the production of NO from different enzymatic and non-enzymatic pathways that includes the NOS, that is nitric oxide synthetase like activity, NR, that is the nitrate reductase activity, and NIR. In this pathway, you can see NO is synthesized from the different pathways, and the main in this is the NR pathway. That is, here you can see nitrate, conversion of the nitrate, then this NO, nitrate from this converted into the NO from the here. So, the production of NO from different enzymatic, all this represented here. Beside this is the xanthine oxidase production pathway, and in the mitochondrial electron transport chain NO is also produced. So, overall it is produced by mainly the enzymatic pathways, and there are some sources of non-enzymatic production of nitric oxides. So, these are the mainly biosynthesis pathways of nitric oxide, and here these pathways are further categorized into two categories depending upon the which type of reaction is going on, and these are the either oxidative pathway or reductive pathway. Most of the enzymatic reaction are categorized under the reductive pathways, and all the different as shown in this one, these are the oxidative pathway, that is NO generated from the either arginine or polyamines or hydrogen. These are the oxidative pathways of NO generation. So, the reductive pathway, this pathway involves the reduction of nitrogen containing compounds to produce NO, and the key enzyme as we have discussed earlier is the nitrate reductase, xanthine oxidase and It also includes in the reductive pathway, that is the non-enzymatic reduction, that is happen simultaneously through the at the acidic condition and through the ascorbate or phenolics. While in the oxidative mechanism, this pathway involves the oxidation of nitrogen containing compounds to generate NO, and the enzyme in this include the NOS pathway as we have discussed earlier, and the polyamine oxidase pathway. So these are the two main mechanism, enzymatic and non-enzymatic, and these are the two pathways from the oxidative and reductive depending upon the reaction, which type of reaction is taking place to generate the NO. So these are the biosynthesis of NO taking place. Then the next one, this is the biosynthesis, and the next for the homeostasis, maintaining the balance of the NO, that is the scavenging is also very important. As I have told you earlier, the balance of NO is very important because at low concentration it incorporates to the cellular signaling, but at higher concentration it can cause the cytotoxicity. So homeostasis is very important, and homeostasis is maintained by the scavenging of different pathways, and in scavenging of the plant is mediated by the several pathways including the phytoglobins, S-nitroglutathione reductase activities, and thyroidexin. These are the three main pathways of scavenging of nitric oxide, and in plants this nitric oxide is scavenged by the also reactive oxygen species and by hemoglobins. So these are the some pathways of nitric oxide scavenging, in which one of the main important is that is a phytoglobin. Phytoglobins are plant hemoglobin that scavenge the nitric oxide. Hemoglobin oxidizes the nitric oxide to nitrate, and the non-chemotic hemoglobin is involved in the oxidative degradation of NO to produce the nitrate in the first case, and in the second one by the second, that is the S-nitroglutathione reductase enzyme, which regulate the S-nitrosylation, that is the post-translation modification mediated by NO. So this thiol binds to NO and transport it to the site of action, and where the nitrothiols release NO after the intercellular diffusion and uptake into the target cells. So this N-nitrothiol glutathione is an NO reservoir and a long distance signaling molecule for the NO homeostasis. So this GS-NO, it also is scavenged as it, and also it is storing it when it requires, it process to the different locations of the cell. And the third mechanism which we have discussed earlier, that is the thyrodoxine H5, which act as a direct denitrosylase and helping to maintain the NO homeostasis. And this equilibrium between the NO synthesis and scavenging mechanism determines the plant endogenous levels of NO, and this homeostasis is necessary for the NO to exhibit its signaling functions. So all the functions of NO depending upon this levels of NO, and it play a crucial role in plant responses to drought, salinity, and heavy metal stress. And this is the outline which we have studied in the biosynthesis and scavenging, that is the total metabolism of nitric oxide in plant cells. Nitric oxide can be generated among others by either nitrate reductase or by L-arginine dependent nitric oxide synthetase. Then NO can react with the reduced glutathione to form N-nitrosoglutathione through a process of S-nitrosylation, and the metabolite can be converted by the enzyme S-glutathione and reductase into oxidized glutathione and ammonia. Then is further this glutathione which make mediated signaling processes, and ultimately the nitric oxide also interact rapidly with the superoxide radical to generate peroxynitrile and a powerful oxygen molecule that can mediate the tyrosine nitration for the proteins. So in this process you can see this is the all over metabolism of nitric oxide, how it is synthesized and how it is mediated signaling responses of the plant, as you can see in this particular figure. So synthesis take place, then its post-translation modification, and then it causes the cell signaling, which is important for the different biological functions of the plants. And it regulates many growth and development processes from growth seed germination to the senescence. So it impart many growth regulation processes. So how this signaling is taking place? So NO, as I have discussed earlier, NO is a very diffusible molecule and reactive. So it can diffuse freely across the cell membrane and interact with various cellular targets including proteins, causing post-translation modifications like S-nitrosylation, and which alters the protein functions and triggers the downstream signaling pathways. So this one is the reaction, that is the S-nitrosylation is very important, that triggers most of the signaling process by the NO. Besides this, NO often interacts with other signaling molecules like reactive species, modulation of calcium signals, protein modifications, and phytohormones, hydrogen sulfide, and creating a complex signaling network that regulate most of the developing process of the plants. So it's not only act as a signaling molecule, but it also crosstalk with other signaling molecules to regulate the different functions of plants from germination to senescence. So NO targets various molecules including enzymes, transcription factors, and DNA that leading to the post-translation modifications that regulate the plant growth, development, and stress responses. So here in this figure, a schematic representation of nitric oxide signaling in plant cells is presented, and that interplay between the nitric oxide, calcium, and protein kinases in plant cells. So the direct NO signaling mechanism, there is a presence of S-nitrosylation, which we have discussed earlier. Beside this, it is also binds with the other metal NO interactions, like NO directly bind to the metal centers in heme containing proteins such as phytoglobin to regulate the homeostasis of NO by converting nitric oxide to nitrate. Then it can also bind to the cytochrome C oxidase to modulate the mitochondrial respiration under the stress condition. Beside this, it can indirectly NO signaling mechanism that interact with the reactive species, that is ROS, in which NO reacts with superoxide to form a peroxynitrite, which modifies and alters the signaling pathways. Beside this, NO that is binds with the cGMP and calcium dependent signaling, in which NO activates the guanylyl cyclase, that leading to the productive of cyclic GMP, which modulates the gene expression and enzyme activity, in which NO increases the intracellular calcium levels and triggering the calcium dependent protein kinases that regulate the various stress and development responses. So NO production occurs through the two enzymatic pathways as we have discussed earlier, and these pathways that lead to the S-nitrosylation, which is also act as a signaling agent in this particular figure. So as we have seen the biosynthesis pathways of NO, so it is very important to discuss what are the sources of NO before discussing what are the roles of NO in growth and regulation, and how we use the NO in other agriculture practices. So if we see the sources of NO, nitric oxide is a crucial signaling molecule as we have discussed earlier. The sources of NO, which are donors which can be used for our research purposes. So NO donors are compound that release NO at are used to study in physiological effects, and in which the most significant and most used donor is the sodium nitroprusside, which is commonly called as SNP, which is one of the most widely used donor in plant studies and releases NO in aqueous solution. So this one is the most studied donor which is used in the physiological studies, that is the SNP. Beside this, that is another donors of nitric oxide like the S-nitroglutathione. This is a natural NO donor that participates in S-nitrosylation of proteins, and diethylene enotate, a rapid NO releasing compound that is used in controlled NO release studies. So these are the some chemical NO donors which we can use in our physiological studies or research purposes. So beside this, suppose you have used NO in your physiological studies, that NO you have to study a specific function, which are the function which is regulated by the NO. So you have to use also the inhibitors, because many functions are linked with other phytohormone signaling molecules. So to detect which function is regulated by the NO, we have to use the inhibitors, because if we use inhibitors, that function if that regulated by the NO, that will not going to happen. So inhibitors is also very important in research practices, because it inhibits the activity of that particular compound, and NO inhibitors which is commonly used as that suppresses the production of NO, like which include the NOS-like inhibitors, because we have seen in the pathways of nitric oxide synthesis, there are the two main pathways of NO synthesis. That first one is the NR pathway and the second one is the NOS pathway. So, the inhibitors like NOS-like inhibitors that inhibit the competitive inhibitors of NO synthetase like activity. So, the enzyme which synthesizes the NO, that is inhibited. So, the function which is related with the NO, that will be not appear in that particular test plant or your. So, these are the some examples here listed here in this particular, which are showing that is the inhibitors. And beside this, there are several scavengers also used for the study in the research functions. That is, the scavengers are those compounds which remove the Na from the sources. So, scavenger neutralizes the excess NO and helping in NO detoxification and regulating its signaling pathways. And the most important in which is used as a carboxyl PTIO, a widely used NO scavenger that react with NO and reducing its bioavailability. So, I am not discussing all of them. You can use sources, and the most common source is sodium nitroprusside, that is commonly known as SNP, and there are the inhibitors like NOS inhibitors and scavengers like PTIO. They scavenge the nitric oxide. So, if you would like to study on the NO in physiological function, you have to provide a donor that releases the NO. You can study it, and if you want to check which function is regulated by the NO, you have to use the inhibitors or scavengers to check that particular process. So, that is why I have discussed this first. Then I am going to discuss about what are the growth and regulation function which is regulated by the nitric oxide. A nitric oxide is a crucial signaling molecule in plants that plays significant role in various physiological and development processes. Its function ranges from seed germination to stress responses, like it promotes seed germination. Here the green one indicates the promoting factors and the red one that is inhibiting, and that both the arrows green and red shows that depending upon the concentration of the nitric oxide and the circumstance of environment. So, it promotes the seed germination, regulates flower development, influences cellular production in roots and prevents chlorophyll degradation. Additionally, NO delays fruit ripening and modulates the lateral and adventitious root formation, and also inhibit the senescence and suppresses programmed cell death. So, all these functions, how NO regulates all these functions. The first is starting from the seed germination, that we will also discuss in the next slide. NO promotes seed germination by breaking seed dormancy and enhancing reactive oxygen species signaling. So, the signaling which is involved in the regulation of NO, it is also involved the crosstalk of AB and gibberellic acid, which we will discuss in the next slide. So, it also promotes the seed germination, then it also promotes the root development. So, NO influences the root architecture by modulating the lateral root formation and root elongation by the interacting the auxin and cytokinin. So, you can see here NO is involving in crosstalk with many hormones as well as the signaling molecules. Like in the seed germination, it is crosstalk with the gibberellic acid and abscisic acid, and for the root development, it has a crosstalk with the auxin and cytokinin. Similarly, for the flowering and reproductive development, NO plays a role in flower transmission and pollen tube growth, and it regulates anther dehiscence, ovule fertilization and seed sets. And beside this, in photosynthesis and chlorophyll synthesis, NO enhances photosynthetic efficiency by regulating chlorophyll biosynthesis and electron transport chain, and it also protects the photosynthetic apparatus under stress conditions. And lastly, the senescence and programmed cell death. NO delays leaf senescence by reducing the oxidative damage and modulating the gene expression, and it also participates in the regulation of programmed cell death under the stress condition. So, these are the some growth and development of processes that is mediated by the nitric oxide. So, these are the some processes, and here I am going to discuss only the one part, that is the seed germination. Here you can see in this figure, for the NO that breaks the seed dormancy, for this it has a crosstalk with the abscisic acid and gibberellic acid. What are the role of gibberellic acid? Anyone can tell? Seed germination through the enzyme alpha amylase. So, functional interaction of NO induces the ABA catabolism and promotes the GA biosynthesis. And this GA biosynthesis that leading to the dormancy, release and germination. And the here arrows and bar indicate the positive and inhibitory effect. You can see here this ABA catabolism is here. This NO is catabolism that regulate the catabolism and it promote the synthesis of gibberellic acid. So, the dormancy is breaking by the nitric oxide through this phenomena and it promote the seed germination. That is why it is very important in the stress condition also, because many of the stress that reduces the percentage of seed germination like salinity or drought. So, as it breaks the seed dormancy, so it also help in the elevating the stress responses like salinity or heavy metal, all these processes which decrease the rate of seed germination. So, I am not going to discuss all the processes in detail. So, now moving toward the next topic, that is the role of nitric oxide in abiotic stress tolerance. So, we have already seen NO is regulating many growth and development processes from seed germination to programmed cell death. And we have seen that seed germination is play a very important role. So, here in this figure, a generalized representation of versatile role played by the NO in plant during various abiotic stress conditions. Like the signaling of NO underpins a multitude of cellular processes that involve in the development and abiotic stress responses in plants. And under stress condition, oxidative stress is generated, which is diminished by the NO by modifying the morphophysiological, biochemical and molecular processes such as functioning of stomata and transpiration under water stress condition, activation of defensive genes and antioxidant enzymes and accumulation of osmoprotectants and crosstalk with different phytohormones. These are the some modifications that is enhances by the NO which help the plant to suppress or alleviate this abiotic stress condition. For example, the presence of salt has been observed to prevent the seed germination and resulting in the significant agricultural losses because it decreases the germination of the seeds. And we have already discussed this, as we know, germination of seed is first stage in the development of angiosperm, and wheat seeds exposed to salt stress can be effectively elevated by the administration of this NO donor like sodium nitroprusside. This is probably due to the enhancement of beta amylase activity, which is directly linked with the seed germination, and during seed germination beta amylase is involved in the degradation of starch and facilitate the conversion of starch into the sugar, which is essential for the proper seed germination. And the mechanism is we have seen earlier in this particular slide ki how NO crosstalk with the abscisic acid and gibberellic acid to promote the seed germination. So, in this way I am giving just one example how NO help in the elevating the salt stress by promoting the seed germination. Similarly, in this figure a mechanism of different stress and how NO can elevate different types of stress. So, NO plays a protective role in drought, temperature extremes and heavy metal stress also. So, it regulates stomatal closure, antioxidant enzyme activities and osmolyte accumulation, and all these things is done by modulating the signaling pathways. So, NO enhances plasma resilience and under the stress conditions, and all these role is done by, so NO interacts with the different target molecules such as mitogen protein kinases that activates the signaling molecules. It also interacts with the cyclic glucosylation monophosphate, cyclic adenosine diphosphorylase and calcium. So, these are the signaling molecules in which NO interacts, and as the protection against oxidative stress, NO can initiate the cascade of signaling event which is listed here and alter the expression of gene and also facilitate the re-establishment of cellular redox homeostasis. So, one example we have seen earlier, and here you can see it regulates number of signaling pathways that initiated by the NO, and in this way it generates a protective mechanism for the plant to withstand against different particular stress condition, either it is a heavy metal or drought. Plant can elevate these conditions during the due to the presence of NO. And here in this diagram, specifically heavy metal stress tolerance has been discussed. So, specifically interaction between heavy metal stress and NO can vary depending on the type of heavy metal, its concentration and duration of exposure and plant species. And as you can see in this particular figure, that NO at low concentration, it maintains the stability of electron transport chain and recovering the process of different PS2 activities, improving the MPK pathway, and also it increasing the different antioxidant defense genes like APX, SOD, GR and CAT, and that maintain in all total cellular homeostasis, as we have discussed earlier, which is very important for the signal transduction, because if the level exceeds, what will happen? It can cause cytotoxicity. So, at low concentration, it maintains this homeostasis that improves the photosynthesis and growth of the plant, that increases the yield. But at higher concentration, that disrupts the cellular homeostasis and ultimately inhibited the photosynthesis and decreased the yield of the plant. So, as we have discussed earlier, its concentration is very important and which is regulated by its homeostasis, which involve the biosynthesis and scavenging of nitric oxide. So, besides abiotic stress tolerance, NO is also involved in biotic stress tolerance, like NO also plays a crucial role in plant defense against pathogen and herbivores, and it induces systematic aqua resistance and enhances the production of antimicrobial compound. For instance, NO has been shown to protect potato leaves from Phytophthora infestans, a devastating pathogen which causes a much loss in the productivity of potato. Additionally, NO enhances herbivore resistance by promoting the synthesis of defense compound like antioxidants or secondary metabolites. So, it is also involved in the biotic stress tolerance. Then the next very important role of nitric oxide, that is in the nutrient use efficiency. So, nutrient use efficiency is essential for sustainable agriculture and it enhances the crop productivity while reducing the excess fertilizer usage and environmental pollution. So, how NO increases the nutrient use efficiency? First, NO use efficiency, like NO enhances the nitrogen uptake and assimilation by modulating the activity of nitrate reductase and glutamine synthetase, which are the two main enzymes of nitrogen metabolism pathway, and S-nitrosylation of NR increases the nitrate reduction to nitrite and promoting the nitrogen metabolism, which is one of the crucial steps for the nitrogen assimilation in the plants. Besides this, it also increases the phosphorus use efficiency, which enhances by it increases the phosphate uptake by inducing the expression of phosphate transporters genes, that is the PST, and NO mediated root system remodeling, like increase the lateral root formation and root hair growth, that improve the phosphorus acquisition from the soil. So, as we have discussed earlier, that NO promotes the root and lateral root growth promotion by the crosstalk of auxin. So, when the lateral root and root hairs growth are increases, so the nutrient use efficiency is already increases, because all the nutrient is absorbed through the root hair with the water. So, when it increases the lateral root growth, it also increases the nutrient use efficiency. Similar way, it also increases the potassium use efficiency, like NO regulate the potassium uptake by modulating the activity of potassium transporters, that is HEC and CUP family, and NO protects plants from K plus deficiency, inducing oxidative stress by activating the antioxidant enzymes. And besides this, iron and micronutrient use efficiency is also increases by nitric oxide, like NO chelates with iron into making it more available for plant uptake and enhances the zinc and copper uptake by a metal transporter genes. And NO also play a role in elevating the iron deficiency chlorosis by increasing the iron mobilizing in roots. So all these nutrients, like we have discussed, nitrogen, phosphorus, potassium, iron, these are the very crucial for the plant growth of development. And NO increases all the efficiency of these nutrients, that is very important for the growth and development of the plants. So in the next, there are some study of case studies of NO, like several case studies have done on the nitric oxide. Here I have listed only few of them. Like in the drought stress, it is a major challenge in wheat which affecting the growth, yield and physiological processes, and nitric oxide play a crucial role in enhancing the drought tolerance by regulating the stomatal closure, antioxidant defense system and osmolyte accumulation and hormone signaling in wheat. So drought tolerance is involved in, it regulate this particular process in several steps. Like first is role of NO in drought tolerance. It first regulated the regulation of stomal movements. So NO interact with abscisic acid to induce the stomatal closure and reducing the water loss. Then it activates the calcium dependent protein kinases which triggers the guard cell signaling and stomatal closure, and enhances the expression of water channel proteins like aquaporins to improve the water retention. Then secondly, it enhances the antioxidant defense mechanism in the drought tolerant wheat plant, like it lead to the enhanced generation of antioxidant like superoxide dismutase, catalase, ascorbate peroxidase and glutathione reductase. And besides this, it is also play an important role in osmotic adjustment and metabolic regulation. Like NO promotes the accumulation of osmolytes like proline, glycine betaine, sugars which help in maintaining cell trigger pressure, and NO modulates this natural metabolism by regulating the nitric reductase and glutamine synthetase, that enhancing the protein synthesis and stress adaptation. So all these processes generally involve the crosstalk of hormones and gene regulation. And beside these wheat drought stress, NO is also enhances the nitrogen use efficiency in rice, like the donor of nitric oxide, sodium nitroprusside, increases the nitrogen uptake and assimilation in rice and releasing the compound that enhance the photosynthetic nitrogen use efficiency, promoting the higher biomass production and improves the nitrogen retention and reduces fertilizer runoff, that contributing to the sustainable agriculture, because it decreases the dependency on the chemical fertilizer, because it directly involving the nitrogen use efficiency in the plants. So it lead to the sustainable agriculture. And in the third example, like we have discussed earlier, that application of nitric oxide as a donor has been shown to reduce the pathogen infection rates, like it enhances the PR gene expression and antioxidant activity and strengthen the cell wall structure, that making plants more resistant. So these are some case studies, and if we see the interaction of nitric oxide with other phytohormones, so we have seen that most of the processes that NO regulates, that is by the doing the crosstalk with the other phytohormones or signaling molecules. Like example, it promotes the root elongation and lateral root initiation through the interaction with auxin. NO also regulates stomatal movement and drought condition by modulating the abscisic acid signaling, and all these interactions highlight the multifaceted role of NO in plant physiology. So in this figure, you can see that showing a different growth regulating processes and what are the crosstalk of NO doing with the different goals. So nitric oxide, as we have discussed, it involved in, like if we see NO and auxin crosstalk, NO promotes the root growth and lateral root formation by enhancing the auxin signaling, and this NO modulates the activity of auxin responsive genes such as pin form proteins which regulate the auxin transport, that enhances the root hair development in response to auxin signaling. So with auxin, what it will do? It will enhance the pin protein that is involved in the auxin transport, and that enhances the root development. If we see the NO and abscisic acid, NO acts as a mediator in ABA-induced stomatal closure and preventing the water loss during the drought stress, And ABA triggers NO production, which activates the calcium dependent protein kinases that leading to the guard cell closures. Like example, NO required for the ABA induced systemic closure in drought conditions. Then if we see the crosstalk between the nitric oxide and cytokinin, NO modulates the cytokinin induced shoot development and leaf senescence, as cytokinin enhances the NO production, which regulate the cell division and differentiation, and NO interacts with CK signaling pathway to control delayed leaf senescence and chlorophyll retention. So, NO delays cytokinin induced leaf senescence and by influencing the antioxidant enzyme activity. With NO also interact with gibberellic acid, as we have discussed earlier in seed germination. NO enhances GA mediated seed germination and stem elongation by modulating the GA biosynthesis enzymes like GA3 oxidase or GA20, promoting the plant growth, and NO also reduces DELA protein stability, allowing GA to promote the cell elongation. Then NO is also have a crosstalk with salicylic acid, and NO and SA work together to activate plant defense responses against pathogen. Both induce systematic acquired resistance by upregulating the pathogenesis related genes, as we have seen in case of a biotic resistance. So, NO enhances SA mediated hypersensitive response and programs cell death in infected tissues. And if we see the crosstalk of NO and jasminic acid, which is essential for biotic acid resistance like insect herbivory. In this case, NO enhances J-induced secondary metabolic production such as alkaloids and flavonoids. So, NO influences J-responsive gene involved in defense and wound healing. So, by this it increases the secondary metabolic production with the J crosstalk and increases the defense in case of a herbivory. And in the last, NO is also have a crosstalk with ethylene and interact with the ethylene to regulate the fruit ripening, senescence and stress responses. In this case, NO inhibits ethylene biosynthesis by S-nitrosylating ACC synthase, a delayed ripening enzyme. So, NO with the crosstalk of ethylene, it delayed the fruit ripening. So, NO delayed the ethylene induced fruit ripening in tomatoes and other fruits. So, you can see it crosstalk with other phytohormone and regulate a number of growth and development processes in plants. Beside this, NO also play a very important role in agriculture. So, the accidental application of NO donors, which we have seen earlier, the most common donor is sodium nitroprusside, has shown promise in improving crop resilience and yield. NO based fertilizer are being explored for their potential to enhance nutrient use efficiency and reduce environmental impact. Additionally, NO has applications in post harvest preservation, extending the shelf life of fruits and vegetables. So, as we have seen earlier discussed that how fruit life is increases because it delayed the fruit ripening by delaying the ethylene hormone. So, in the next one it also involved in some other activities like NO aids in the plant microbe interaction, promoting nitrogen fixation and microbial association. It also have a potential tool for pest management and fungal and bacterial disease control. Beside this, NO also enhances the crop adaptability to climate change and hydroponic systems. And NO also influences the production of phytochemicals which contribute to the plant defense and human health. As we have seen in the case of a herbivory, it interacts with the jasminic acid and it increases the production of secondary metabolite that is crucial for the in case of a plant defense in case of a herbivory. Then it has an application in plant tissue culture that aiding the micropropagation and regeneration. Then it is also involved in the breeding strategies are exploring NO responsive traits for the crop improvement. So, these are some sections in agriculture where it is used for the production, uses in the agriculture for the better growth and productivity. And this figure showing all the details which I have discussed in this particular lecture from biosynthesis to the role of nitric oxide. As you can see here, this showing the biosynthesis of NO, Then this NO, how it is scavenged, then its post-translation modifications that is mentioned here. So if we see NO, as we have discussed earlier, synthesized by the enzyme that is nitric reductase and NOS, and from the different polyamine pathways. Then by the signaling it increases the antioxidant defense system, and how when this antioxidant defense system is increases, the overall plant of the growth is increases under the stress as well as in the normal condition. So this diagram depicting the overall growth of the plant. So if we see the what are the challenges and future perspectives. So despite its potential, there are challenges in the precise regulation of NO levels and understanding its crosstalk with other signaling pathways. Although we have seen here many crosstalk, but still many pathways are also missing. So future research should be focused on developing NO-based agrochemicals and exploring its role in climate resilient crops. And these efforts could lead to the more sustainable agriculture practices and improved crop productivity. So in conclusion, NO is a versatile molecule with significant role in plants' growth, stress tolerance and nutrient use efficiency. Its potential application in agriculture are vast, ranging from improving crop resilience to enhancing nutrient uptake and reducing fertilizer dependency. So by harnessing the power of NO, we can move toward the more sustainable and productive agriculture system. So that is, thank you.